अब ये काम कर रहा है, परफेक्ट। थैंक्स। So, guys, am I audible till the last row? Thanks.
Yeah, I'll just wait for people. आज ठंड थोड़ी ज्यादा है. Okay, actually, I'll start the procedure and then we can have Vishal's talk. So it's a pleasure to welcome Professor Vishal Rai from IISER Bhopal, who right now he is the professor and head of the chemical sciences program there. So Vishal and I have a long, we go back a long way with the last 10 years, 10, 14 years. We both started the same time pretty much, and we have been sort of meeting each other in meetings. So it's been a pleasure to host him for the second time in TIFR in all these years. Vishal actually, as you have seen from the abstract, is a precision protein labeling person. So this is something that he will explain what it entails and what kind of chemistry brings about selectivity of labeling proteins and why it is important. But we will give a little bit of a background to where he comes from, how he thought about all these things that he does now, where did that all originate. So Vishal actually did his PhD not very far from this place, from IIT Bombay actually, with Professor Nambutri. And this was in the period of 2003 to 2008. And he subsequently went for a postdoctoral position at University of Toronto with Andrei Yudin. This was in the period of 2008 to 2011, 3 years of postdoc. And that's where he sort of made some very seminal contributions to use of peptide macrocycles. And that turned out to be part of a company that started in University of Toronto called N-Cycle Therapeutics. After his return to India, he directly joined IISER Bhopal. Of course, Bhopal was 3 years old at that time, IISER. And of course, with a new program, starting a new program in a department that is still setting its legs up, it's a big challenge. But hearing all the years from Vishal how things have evolved, right now Bhopal is doing amazingly well. Some very nice chemistry is coming out from the department, and Vishal is actually one of the front leaders in that. Vishal's research currently is trying to develop chemical technologies for precision engineering of proteins. They are also involved in synthesizing homogeneous antibody conjugates, protein immobilization, and analytical tools for peptides and proteins. And his research team actually sort of has an aim to apply the science that they're developing in the lab for societal benefit. And so he has actually founded a company. He's a founder and director of Plebeltech Private Limited, with state-of-the-art protein and antibody engineering technologies that they're developing. They are sort of now entering into the market. And he may talk about some of these efforts, I don't know, but I've had very nice discussions even today. Because of this very highly controlled chemistry, Vishal is has won lots of recognition. There are lots and lots to read from this list, but thing that I wanted to mention is last year he was awarded the Rashtriya Vigyan Puraskar, which is the Shanti Swarup Bhatnagar Prize. So with that, Vishal, I welcome you to the floor and
Thanks.
share the work.
Thanks a lot. Okay. At the onset, let me thank JD for this kind introduction, and both JD and Deepa for inviting me back to TIFR. It's always a pleasure to be here. And as he pointed out, while it's my second trip as a faculty member, I was a frequent visitor to your NMR facility, specifically when we had to run 2D NMR. We had some really good friends in Professor Chari's and Professor Huzur's lab who would do it for us. So we used to quite often come over here, specifically in the weekends, because that was the time when the instruments and the labs were less occupied. Now, for today, I thought I would share one of the philosophy on how human behavior has been driving the research program that we have been running in our lab for almost last 13 to 14 years. And it would not be a stretch if I tell you that the human behavior has actually inspired most of the technologies that we have developed in perspective of precision engineering of proteins. But before going into any of those details, let's just ask the most basic question that might come to your mind, that why do we even need protein engineering methods, right? And if we will try to understand that, it will become slowly obvious that why chemical methods would be playing an important role in meeting the technological demands that comes into this perspective. So the very basic knowledge of how we can regulate the precision of making bonds with the proteins are absolutely essential for developing tools for biophysical chemistry or biochemistry or even diagnostics. Now, what you would realize that this segment contributes to more than 6 billion US dollars today, just the segment of protein and antibody labeling with the non-specific technologies. And this also happens to be one of the most rapidly growing segment within the biotechnology sector. But what really excites our research group is the possibility to contribute towards directed cancer chemotherapeutics. And that we want to do by synthesizing highly pure homogeneous antibody drug conjugates, or it can be abbreviated to ADCs. So let me just take a couple of minutes to define what this is and what this does. As the name suggests, this antibody drug conjugates has antibody and a drug or a toxin molecule. Now, it provides a state-of-the-art technology today for tackling directed cancer chemotherapeutics. And the reason why it can do this is because of the specificity of the antibody, which is something that can target an antigen that is a signature that is present in the tumor cells. And this signature can be targeted very specifically with the antibodies. The only thing is they are not great in killing. So what we need to do is that we have to take the best killers in the business and couple it with those antibodies. So when you have both these things combined together and this is given to the patient, the antibody can go and specifically target the signatures that are there on the tumor. And eventually, this overall complex is going to be degraded by the cellular machinery in a way that the toxins can be released, leading to killing the tumor cells specifically without harming the normal cells, as you would anticipate. Now, in the last 2 decades, we already have the first 10 drugs that are there in the market. And if you have a close look in this whole portfolio, you would realize that these molecules or drugs are sold as heterogeneous mixture. So they're not chemically pure. And what happens is that there will be 0 to n number of these drug or toxin molecules that would be attached to the antibody. And this heterogeneous mixture is there because nobody knows how to control the selectivity of that bond formation. And it is also very well accepted across the fraternity that this heterogeneity is quite harmful or detrimental to various pharmacokinetics parameters. And at the same time, it makes it really difficult to get batch-to-batch reproducibility. Randomness is tough to reproduce, whether it is chemistry or whether it is us in terms of our behavior is the exact same situation. If you have random outcomes, it is not going to be straightforward to reproduce. And that is something that most of the companies across, yes.
These samples when you produce, then how you can use to the patient?
That's excellent question. That is one of the reasons why many of these ADCs are still not used as the first line of therapeutics. People would first go for the antibodies alone. And when the patients would develop resistance in certain cases, then when these antibodies alone are not working, then only when it comes down to the critical situation, the second line of therapeutics would have ADCs. So you would imagine that if this problem can be resolved, then ADCs probably would also qualify as first line of therapeutics. But thanks for asking that question. Yes.
What's the N that you mentioned? What's the typical value of N that you mentioned? 0 to N
Now, depending
toxins.
on, so let me just ask your background and then I'll probably answer accordingly. Chemistry, biology, physics, physics. Okay. So depending on the residue that we are targeting, in different drugs, people go mostly 8 of them would probably target just either lysine or cysteine. If it is targeting lysine, then in that case, it would, the number of drugs that you would notice in the mass spectrometry data would be somewhere between 0 to 16, with average coming down somewhere around 8. That would mean, though, if you will sequence this whole thing, you will realize that up to 78 to 82 lysines would be heterogeneously involved in the connection with that particular drug. If it is targeting cysteine, antibodies don't have free cysteines. So they will have to reduce the disulfides to create those thiol groups. A functional group that is then utilized for installation of drug. In these cases, the average loading would be around 4. The heterogeneity will be there. And you would know that after reducing that bridge that was holding the structure of the antibody structurally would not look the same. So each of these dimensions have different type of problems associated with it. But in general, most of the drugs that target lysine, the answer is the range, average loading would be somewhere between 4 to 8. Total number would be ranging somewhere between 0 to 16. The number of residues involved would be somewhere between, if thiol, around 8, if lysine, then up to 82. So that's the level of heterogeneity we have to take away. Okay. Now, so now I hope that I was able to convince you in this perspective that it was really important that, one can address these challenges associated to the selectivity to cater to this requirement. And it doesn't matter if you're interested in ADCs or not, even if you're using any vaccines from the market, you would realize that conjugate vaccines constitute almost 25% of the overall vaccine market. And it's just that instead of antibody, now you have a carrier protein, and instead of toxin, you have a particular antigenic compound that is associated to it through a bond formation. But then the problems of the selectivity and heterogeneity remains exactly the same. And the same extends to the antibody-based imaging-guided oncologic surgery or photodynamic therapy. Now, we have been working on some of these problems along with our collaborators over past several years. But while we are looking into protein-based therapeutics and some related solutions, our academic part of the lab is really trying to see that in long run, would it be possible for us ever to go after a single site of a single protein inside a human body? Because if we can achieve that, then we are talking about getting the specificity that today can be imagined only with biomolecules such as antibodies. And that would be exceptionally interesting, because then we can cut down the cost of precision therapeutics to the level where we can probably provide it to a very large segment of the population across the globe. If you're not aware, most of the biopharmaceuticals are going to be very expensive, specifically if you're talking about the antibody drug conjugates. But before we can work with complex systems such as humans or tissues or even cells, we would first need the knowledge of how we can regulate the basic principles and parameters to define selectivity in isolated proteins. And once we have that knowledge, then we can consider translating it from one layer of the complexity to the other, while understanding what additional attributes are pushed forward by each layer of the complexity that we will be dealing with. Now, at this point, a very good question to ask would be that, do we not already have drugs that are highly specific in nature, right? Or in other words, how selective are the most selective small molecule-based drugs? So I thought I'll just toss up a multiple choice question for you and see what options do you choose. If we take a drug that is supposed to target, let's say, an enzyme, which is a protein, how many proteins do you think the most selective drugs might be targeting? Around 10. That's option B. Any other options? 1. That's something that we would love to see one day. Yes. Option D. Okay. C is left. Any takers for C? Okay. So, keep these thoughts in your mind, because this is a pressing question that a lot of people in the community have been asking. And they have been, from the physics and engineering side, they have been trying to develop analytical tools to look into how we can develop data with low concentration samples with high confidence, so that we can know exactly what is happening. But I'll just give you one example in this perspective. Kinases are probably one of the most well-explored enzymes, simply because they play a critical role in the regulation of biological pathways inside us. And that's the reason why it's not really a surprise that for past several decades, they have been investigated heavily, and they have led to multiple approved drugs. Maybe more than 38 drugs in just last 15 years got approved in this segment. So if you look into what number of targets the most specific kinase inhibitors can go after, that number lies somewhere between 30 to more than 100 known targets. And then there are many unknown ones that you cannot identify through the chemoproteomics workflow that is typically utilized in such studies. So we hope that in the long run, we would be able to address some of these challenges and hopefully resolve the issues related to the side effects that are currently observed in the patients who administer these drugs. So with this, yes, please.
Kinases themselves really broad class of protein molecules, very diverse.
Yes.
So is it that the drug is really developed for certain specific kinase and then going to 100 kinases in human body, or?
Very good question. So typically, if you are looking at an assay, depending on how much money you invest, with higher money, you can get up to an assay for 520 catalytic domains for kinases, representing 520 kinases from the kinome. Now, you would, okay, almost all the effort that has been towards the kinase inhibitor development will be targeting the ATP binding site. And The problem is, if you look into the structural similarity of all these kinases, it's just so similar to each other.
Exactly.
So I can imagine why it would be a nightmare for people to actually try to get selectivity in targeting one of them. So even if, let's say, a particular kinase is overexpressed in a certain type of disease state, the problem is that you will not be touching only that. So in the other non-disease cells, you would still have the normal level of expression of other kinases, and this drug molecule would do equally good job in stopping them from doing their job.
Exactly.
So
So that's exactly the point, because kinases are very broad and you have many different. For example, let's say if you take ion channels, then you won't have much diversity, right? So there, I would be surprised if you say 100 targets for a given developed drug.
if you're targeting ion channels per se, yes. So, same thing when it comes down to, very specific mutations that you are probably targeting, you can argue that you can develop a particular, let's say, inhibitor or a binder or modulator for that. And that is true. At the same time, the numbers, when you look into how many non-specific targets within the proteome it goes for, that will still remain pretty high. So I can definitely talk about some of the other examples that we pursue looking into regularly on how the field is developing. So now with this in background, we started somewhere around 2011 with a very basic question that let's see that would it be possible to develop methods or chemical methods that can allow us to engineer bonds very specifically with protein. But more likely at that time, we were thinking that let's just first see why people believe it won't be possible. And can we understand the physical organic chemistry part, the mechanistic aspect of how this is regulated, what contributes to non-specificity, and is there a window for deconvolution, right? And can that reactivity be resolved further? So I will just take few minutes to explain three terms that would be jargons. After that, I will not be using any other jargon, but these three are important. So I thought I would explain that to you to this. In fact, considering that we also have people from very different backgrounds, right? So proteins, the way we see it is, it's organic molecule for us. Yeah, I know it's a biological molecule, but let's just consider it's organic molecule. And it is full of nucleophilic residues. Nucleophiles, by definition, are handles that can donate a pair of electron. So if something can donate a pair of electron and I want to form a bond, I will add an electrophile that can accept that pair of electron, right? And then the bond is formed. So it's as simple as that. Proteins with bunch of nucleophilic groups can donate a pair of electron from multiple sites. And if I have to do a bond formation, I'll add an electrophile that can accept that pair of electron, make this covalent bond with the protein. Now, if I take this protein in solution, add an electrophile, you would realize soon that there are multiple types of groups that are present that can donate that pair of electron. Obviously, their donating capabilities are going to be little different from each other, but there will be blue ones which will compete with the red ones and the orange ones and so on and so forth, right? So now if I have an electrophile that can differentiate the blue one from the other colors, then we will call it chemoselective. So chemoselectivity is differentiating one type of functional group from the others. And let's say this particular electrophile can differentiate that blue functionality, which is amine. In proteins, it is called lysine residue. Then in that case, it will soon realize that there are multiple copies of this blue residues. So for single site modification, it will have to differentiate one copy from the others. And that is what I'm going to term as site selectivity. Okay. So for single site modification, I need a combination of chemoselectivity and site selectivity. Now, proteins also have their own personality. For example, if you look into just these three blue groups, this particular one offers higher solvent accessibility. And, it can also, under given physiological condition, let's just assume that it also offers higher reactivity. So it will give you the best combination of reactivity and accessibility. So when an electrophile is added that is selective for blue groups, this would be the first one to react. Maybe after 5%, 6% labeling, others would start participating, but this would be the first one to react. We call such centers as reactivity hotspots. And that also raises another question, that what if we want to modify this one or this one without touching anything else, right? Would it be possible to beat the inherent reactivity order of the protein and come up with something that is modular? So one more jargon is modularity, and that's more or less it. Now, if just imagine that this protein is present not alone, but in a milieu of thousands of biomolecules, then you would also have the question of protein selectivity along with the other selectivity parameters that we discussed. So with this little background, you would also notice that not all the blue residues had the same social ecosystem, which means that the social ecosystem of each residue is going to create a spectrum of behavior for those residues. And now that might not be a very bad thing. It just adds to the complexity a little bit more for developing a precision toolbox. But if you think about it, the social ecosystem is something that is probably the first thing that we start learning about after we are born. So while it is a complex system, it also offers us enough number of variables to serve as a model for protein engineering problem, which also offers very high number of variables, right? So if you think from the basic algebra perspective, it's not a problem to have more number of equations than the number of variables, but other way you would not be able to solve all the variables, right? So that is what we thought. And I'll just give you one example of how this works for us. Imagine having a person who is in two different environments with different interacting partners. Now, I do not know if the students in the back are imagining this as lab and this as Friday evening place. But my point is simple, right? That you do realize that different interacting partners and different environment can really make a difference in terms of what behavioral profile comes out of a person, right? So you can draw inspiration from that and argue that the same social ecosystem deviation would actually be helpful, because then you can regulate the interacting partners. And if you have control over that, then you can actually utilize this as a powerful tool for developing a comprehensive platform for precision engineering of proteins. So with this little background, we started with the simplest question we could have asked, that let's first see if it is possible for chemical methods for targeting the reactivity hotspots in the proteins to a level that it is practically useful. We do not want 5 to 10% conversion. Let's see if we can really get some meaningful conversions. And we realized that if you can regulate some physical organic chemistry parameters, play with what rate determining a step is and how it is dependent on concentration and whatnot, it is actually possible to target certain reactivity hotspots with very high specificity. Now, different proteins in this case behave a little differently from each other, as you would expect. So this technology is not as broadly applicable. It requires re-optimization every time you are working with a new protein, right? And then we made this question a little bit more complex after learning this aspect and tried to see if we can differentiate the reactivity of a residue from its multiple copies that are present in the protein. And we learned that not only this is possible, we can actually translate it to a structurally diverse set of proteins with some of the very complex residues such as lysine or histidine. And the reason why I'm saying these are complex is because they are very high frequency residues, so many in numbers in many proteins, and they love to be on surface. They are mostly positively charged, which means their solvent accessibility and coordination with solvent would always be higher. besides, what we learned is that you can install handles that you can use for bioorthogonal and chemically orthogonal transformations, and you can play around with these things. But till now, we were still dealing with reactivity hotspots or the residues that were not far in terms of reactivity from the reactivity hotspots. So while this technological platform till this point was version 1.0 for precision engineering, it still required a lot of work beyond this point to see if we can have a control over what we want to modify. So the question was very simple. Can we have modular methods? Can we go beyond the protein-defined reactivity order? Can we switch the site of modification while doing that precision engineering? And while looking into that, well, the most obvious challenge was not one, a combination of challenge. So the point was that if I have to deal with, let's say, let me pick 3 young people. Let's say if I'm dealing with these 3 people together, right? And I have to fight against them. Obviously, I'm going to lose the battle. The chances are very high that they are going to beat me up, right? But what will be my strategy if I have to deal with them? I'll probably run away from them, see that they run differently from each other, so I can segregate them out, right? And put them into different places. And now I have an opportunity to tackle each of them one at a time, which also means that I should be able to understand each of them better and devise my strategy according to each of them, right? And that probably would make my job easier. That is exactly what was required for solving the problem of modularity when we were looking at a protein and had a combination of challenges that needed to be separated from each other or disintegrated. So a theory that we coined at that point was a disintegration theory that argued that, well, if we can segregate all these challenges, we have a better control over what we want to do with them. The problem, though, was that how do we really disintegrate these challenges, right? Because one of the problems that we realized very soon was that the organic chemistry that was known or synthetic chemistry that was known by that time was not giving us enough options to utilize certain tools for disintegrating these challenges very efficiently. So this forced us to look into how organic chemistry itself evolved over a couple of centuries. And when we realized that it was primarily the growth of chemical industries that was responsible for all the organic chemistry that you practically read in your textbooks, right? Now, when the chemical industries were growing, one main focus at that time was scalability. And can you do bulk production of these chemicals? Which also means you want stable compounds. You do not want to make 1,000 kilos that will go bad over time, right? So it was quite obvious that you would focus on irreversible pathways, which means if starting materials went into the product, it should not come back under normal conditions as long as possible, right? Now, if you look into our own body, you would realize that it works in a completely different way. Most of the transformations are regulated by reversible pathways, where if something can go forward, there will be an option to come back. Now, that can be a dynamic equilibrium, or it can be stimulated by one factor or the other, right? And you would soon realize that many biological pathways are practically multi-equilibria systems, right? So at this point, we argued that it would be great to see if we can learn from our own body and utilize these reversible pathways to see if we can deconvolute the challenges that I just talked about. Now, you would realize that one great power that the biological system gets with this is the capability to select, adopt, evolve, right? The capability of evolution is pretty much defined by the fact that it uses reversible pathways. It can make something, and if it doesn't work well, it can come back and try to make something else, right? So the obvious point was that so far we were trying to use this irreversible pathway to address both chemoselectivity and selectivity, and we are not doing very good in terms of what we really wanted to get out of it. And then we decided that let's just use this reversible pathway, disintegrate chemoselectivity from site selectivity, and now we have control over how we want to regulate chemoselectivity or how we want to regulate the site selectivity. We can also use more than one reversible steps, and that basically opens up a scope for exploring selectivity profiles that were not imagined till a certain point of time. I'll give you just one example from this particular pathway in terms of what it leads to, what the end product looks like. So it led to something called GlideTag technology, where we use two reversible steps followed by an irreversible closure. And the GlideTag technology allows us to make a very specific carbon-carbon bond at the N-terminus glycine residue of a protein. It does not touch any internal residue. It also does not touch any other N-terminus residue. So you can work with isolated protein, you can work with milieu of proteins, and it would still be very specific towards that one particular residue. Now, if there are biologists in audience, and if you have used sort tagging, you'll probably also see this as an artificial small molecule enzyme that's probably more effective than the actual enzyme itself in terms of specificity, because it needs only one single glycine for doing that specific modification, whereas the actual enzyme requires a sequence of 5 glycine residues, one after the other, to do a similar job in terms of modification. Later, we also learned how to break bonds, and that worked out pretty well. And right now, some of our biology colleagues are using this GlideTag for doing metal-free protein purification for recombinant proteins. Yes, Ampana.
You have to install glycine in the N-terminus and then you achieve the selectivity.
Perfect. Thanks for asking this question. So normally, okay, when we were doing this work, we had no idea about how much would be N-glyproteome. There was not even a single paper talking about it. But then at the same time, at that time when we published, at that time we were putting a tag. We had a precision protease that would cleave off the tag, leaving N-terminus glycine over there, and that glycine is what we were capturing. Later, we learned that you can take a cell lysate, you can try to target the whole cell lysate, enrich the sample, and then you would be able to see that there is N-glyproteome. The only problem, though, is this N-glyproteome or the proteins that have N-terminus glycine residue is something that is not translated as such, right? So it will always have methionine at the N-terminus. It's just that if the second amino acid is glycine, there is an enzyme that cleaves off the methionine, leaving glycine at the end. This glycine, though, does not remain there forever. It's not stable for protein. So either it will undergo a post-translational modification, regulating the downstream consequence of that N-glyprotein, or it will take it to the degradation pathway. And then there are a few other pathways that are associated with such transformations. But N-gly does not prefer to stay as N-gly for a very long time. No. So typically, if it is for recombinant proteins, it's a separate situation. In recombinant proteins, you can attach tags, which can be easily cleaved off with 2-3 different enzymes to leave N-terminus glycine, and you can capture and enrich and do late-stage modifications also if you want to. Or if you want just native N-gly, that is also easy to cleave it off. For example, insulin would have N-terminus glycine, right? So and that is stable with N-terminus glycine. But when it comes down to targeting something within the live cell, then it's a different story. Then you are not looking at any recombinant expression. You're looking at just the native cell. And then we now have chemistries that we don't talk about yet because it's still maturing. We do have reagents that would target a specific protein, regulate what happens to it and where it goes eventually and how it regulates the signaling pathway. Now, whether it is specific to just one protein or not, we cannot tell you at this point. Maybe another 3-4 months, I'll be able to give answer to that question as well. But have I answered your complete question? Okay. Now, we were talking about using the reversible steps for deconvoluting these challenges. In the same way, you can actually use 2 reversible steps and deconvolute chemoselectivity as well. Don't worry about the chemical reactions there. The whole point is, if you can use 2 reversible steps to generate a protein that can react, another protein that can react, this generates a nucleophilic intermediate, this generates an electrophilic intermediate, but both are reversible, then you can actually have a covalent irreversible modification only if these 2 proteins interact with each other. So we can actually deploy protein-protein interaction to regulate site selectivity by deconvoluting the chemoselectivity with 2 reversible steps. And that you can imagine why this could be powerful. The way where our dream is going in this direction is, it would be great to have molecules that just sit there as silent watchers and make some reversible intermediates, but do not harm or change any property. But if there is a certain protein-protein interaction that is disturbing the whole homeostasis or the balance of the cell, then they start doing the modification and inhibit that protein-protein interaction. We are yet to find a successful model, though, to solve this or translate this in terms of functionality. But
or you have actually translated this into some
so
real
it's not in cells.
It's not in cells
yeah.
yet.
So in vitro, it works great. It gives
Okay.
you precise modification, and we can identify the intermediates where both the proteins are involved and so on and so forth. But what would be fun is to do it in the cell. I just wish we have 2 more human resource to take up this challenge. That is where we are right now limited, because everyone in the group is so occupied with a bunch of exciting things that they're doing. Now, coming to the next part, again, don't look at the structures. The simple thing, we can use 2 reversible steps again for deconvoluting chemoselectivity and site selectivity. And that opens a completely new targetome for precise engineering that was not accessible earlier within this community. So I hope that with this, you would have agreed on the fact that this deconvolution or disintegration of the challenges does work well. And it was time for us to test it for the next stage and see if this disintegrate theory that's inspired by the human behavior can allow us to develop a modular technology. Yes.
Sorry. Just in the previous slide,
Yes.
do you do all these reactions in pure water?
No,
Or-
it's in buffer.
Okay, buffer,
Yes.
but it's just water, no other solvent, right?
Okay, depending on the reagent, it could have 1% to
DMSO.
1 to 10% DMSO.
Oh, so you use the DMSO to put it in for a reaction.
So some of the reagents are not soluble in the required concentration in water.
Right.
So we will have to put some DMSO
Sure.
for the solubility factor. And that's another thing we can discuss later,
Yeah, sure.
that
Okay.
DMSO can probably play a role as
Yeah.
well. Yes. Yes. Oh, okay, sure. So, okay, non-chemists, close your ears for 1 minute. So this particular aldehyde, which has hydroxyl in the proximity, that generates 1-imine. This aldehyde with no hydroxyl in the proximity also generates an imine. The only thing is that this hydroxyl group, if you look into the secondary orbital overlaps that it provides, it makes this imine non-electrophilic. So you cannot add an additional nucleophile to it. It will just not work. It's highly unreactive. This aldehyde, on the other hand, would form an imine that can react with the external nucleophile. So the first step is this aldehyde adding chemoselectively to all the lysines, whereas this aldehyde will go and form imine on the proximal one. And when you will add external nucleophile, it can only add to the imine formed by this, but not by this. So first is chemoselective step, second is site selective step. It will react with most of the solvent accessible lysine except the ones that are blocked by a proximal one to this particular aldehyde. So instead of intermolecular reaction happening and forming that imine, the intramolecular imine is going to be formed faster. So intramolecular pathways would be having kinetic advantage. Yes, and if you do not have another lysine in the proximity, then it is the other one is, let's say if it's little far, is still free to make the first imine. There are times when they are less accessible, but if you look into the structure of most of the protein and the sites that we are modifying, they don't look too much different in terms of accessibility. So I think that there is still a lot of a scope of having the mechanistic underpinnings sorted for these type of problems. There are variables that we are still not imagining. I can talk more about this afterwards, where one of our collaborators, Suman, he helped us understand few variables that nobody would have imagined would work that way. But I'll come to that point once I talk about the LDM technology.
Yeah. So when we talk about the accessibility in a
Hmm.
protein of a residue,
Yes.
then what does it exactly mean? Because it really depends on the reactivity of the reagents, right? Based on
Not
what you are
always.
judging.
No. So normally when we define reactivity of residues, you can, one way you would define as a chemist is pKa values. For example, if you take the most reactive lysine in a protein, it will be buried inside a catalytic domain and your reagent cannot even go there. So if it cannot go there over a period of time, it doesn't matter, it's very reactive. The one on the surface that's less reactive would still react. So that's why when it comes to reactivity order, it is not defined only by the pKa value of the residues, but a
I'm
combination
not saying
of
the reactivity order. I'm
Yes.
saying the accessibility order. How does
access.
one define the relative accessibility of a particular residue in a protein?
You cannot. The way people define it is by doing a non-selective modification and seeing in which order things are getting modified. The other way that some of our
Then this is a so this is a property of the reagent itself, right? Because different reagents have different
in that
reactivities.
case, you would have to imagine that your reagent is chemoselective and does not differentiate residues based on their local microenvironment. That's an assumption. Yes. And if you are an NMR expert, then in that case, you will probably, or mass spectrometry expert, then you will probably also do deuterium exchange experiments to look into some of those accessibility problems. Okay, so now moving to the next point, how do we come up with a modular technology? So let me just give you a quick recap of the philosophy that we discussed earlier, where we argued that a person placed into different environments with different interacting partners should be able to display a completely unique behavior profile. So the point was, how do we regulate these interacting partners, right? So if you have to think in this particular audience itself, if you have to identify male members and your shortlisting criteria is that find a male member in this audience, right? You would have multiple options, right? Imagine if I give you another shortlisting criteria, one more filter, that find a male member who is placed at a defined distance, at a defined direction from a female member. Now you would realize that this would allow you to limit the number of pairs of residues or pairs of people who would be participating here. So the trick is very simple. Can we target a pair of people? Can we increase that number of filters, right? And that is exactly what we thought of doing in this perspective. So if I have to engage the social ecosystem, I will have to target a pair of residues instead of one in a given order. So in this case, I have highlighted two residues. One is blue, the other one is red. For people who understand this is lysine and histidine, but blue and red would do the job. The point was that we will still use a reversible reaction first to disintegrate the chemoselectivity and place the reagent on all the solvent accessible blue residues or the lysines. Now we can control the position of another reaction handle near the red residue depending on how we design the spacer. Now, a basic chemistry part, the intramolecular pathways are always going to be kinetically superior by few orders in comparison to any non-selective intermolecular pathway, right? So this gives you an advantage. But what is very important to see here is that now you have a control parameter in your hands. You can design this spacer to dictate the position of this reaction handle near this red block, or you can change this design in a way that this reaction handle would be near this particular red block and not this one, right? And that is the synthetic capability that you have in your hands. So you can regulate these aspects. And that's exactly how it works. And in this,
what it is presenting at what time, because these are fluctuating structures. You still have to have a good knowledge of the structure, right? Or even the excursions from the structure.
that's a very good point. So the argument is that if we do not know the structure, how do we actually go about it? So if you really want, if let's say the research question is that, hey, I want to modify a specific residue number X. In that case, yes, you need the structural knowledge. But if you want to first check out the point whether this would be possible or not, then you can design a library of reagents, because what you would notice is that the first reaction is just chemoselective. It does not differentiate between different lysines, right? And it is a good thing because it means that it does not depend on the structure of the protein. It will just go at every lysine that is accessible and will form the linchpins there. And then it is completely in your control of how you are designing this. So there are two aspects to it. One, you can say that, hey, let's design a library where I have different length of linkers or spacers, and I will see which one works. But the ideal thing would be that I can see the structure and I can say, okay, this is exactly what we will design, and this will target this one. So if you as an end user ask me, Vishal, give me a reagent that will modify lysine number this or histidine number this, can I provide it to you without doing any screening? We are not yet there. We can do that with 50% confidence. That too, thanks to Suman, that we understand this a little better. And I can explain this later. The technical part, I will explain this later to you on this matter. So now, if you look into a protein structure, what it literally means is that I can take this red block or this red block and modify each of them selectively simply by how I design the spacer that we were talking about. So we translated this to a huge set of proteins, and we were able to create a nice library of reagents where we can use a few different residues as linchpin points and then target a complementary end target, right? This could be histidine, could be lysine, could be carboxylate, and a few other residues. Now, I'll just give you one representative example to tell what it means. This was with insulin, specifically human insulin, and we applied these different technologies to show that you can modify these different residues in very different domains with complete precision on the single site specificity, which also means that now we have access to synthetically engineer insulin and utilize it for various purposes. So one of the translational aspects that our startup is looking into is, along with Eli Lilly, we are trying to see if one specific engineered insulin out of this list could be a source of next generation insulin-based therapeutics for them. Now, at this point, we were all set to see if we can make antibody drug conjugates with this technological platform, which we called as linchpin directed modification, or LDM. But before getting into those details, let me just give you a quick recap of what challenges we discussed about it. And we looked at the antibody drug conjugate structures where we said that, okay, it requires 3 components. The first one would be a toxin, which in this particular example is monomethyl auristatin E. Don't bother, is the killer. So the orange stuff is the killer. The purple one is the one that was required for a specific targeting. So this Y-shaped structure is typically what you would show the antibody with. And then you need the linker to connect the two. And that's the time when you have to ask the question about homogeneity. And once you have controlled this selectivity that would lead to homogeneity, and these ADCs are injected to the patient's body, it's very, very important, as you would realize, that making bond is just one part of it. This bond should be strong enough, because if the bond strength is not up to the mark, then this is going to be released before even reaching the tumor. And the stability in this whole delivery pathway is going to play a very critical role. And you would imagine that if it is released, then the non-specific targeting of the cells is going to be a real problem. So these problems, once they are addressed and you know how to control the bond strength and what bonds you would be using, these antibody drug conjugates would eventually reach these signatures that we talked about, which is called antigen, and they are going to bind. So notice that you cannot affect these things while the conjugation. This is one of the problems why the current market drugs also have issues in terms of efficacy. And once you have addressed those things, these overall complex goes inside the cell, and eventually there is a cellular machinery which would make sure that these toxins are going to be released, and then they will make sure that the cells on the tumor tissue is killed. Now, this whole pathway that we saw in the video is defined here in pathway A. We will skip the details. The only point that I want to show in this slide is that there are more pathways that are engaged in this overall activity. And there is enough clinical data for the first generation ADC that is now available. And if you look carefully into all those aspects, one thing becomes very clear, that an ideal platform for constructing these ADCs is not necessarily required only for regulating the homogeneity or drug to antibody ratio. It must give you a very good control over the residue that you will be using, which will define the bond strengths that you will be using, and then flexibility with the linkers and the conjugation sites. Now, that is exactly what we are trying to do with the IDM platform. So now I'll just give you one representative example from that library that we have created so far. We took trastuzumab, which is probably one of the most common antibodies that is used in the current day scenario and targets a specific type of breast cancer. And then we took emtansine as one of the toxins, which is again a selection because we wanted to do a head-to-head comparison with Kadcyla, which is one of the commercial ADC from Roche at this time. So we realized that this homogeneously engineered antibody drug conjugate from the LDM platform does a pretty good job in inhibiting or killing the tumor type cells without touching the normal type cells at all. And it outcompetes the Kadcyla in practically every initial trial that we have done so far. But the bond that we have engineered here is not ideal for all type of tumors that we want to go after. So we changed this platform using its modularity a little and targeted the lysine residues, which I earlier depicted as those blue spots, and demonstrated that now we can have very high bond strength in this perspective. And we still have ADCs that are very precisely engineered, and they can go for killing the tumor type cells without actually hurting the normal type cells. What we learned beyond that is the overall loading of the toxin that is there in the commercial drugs. You can actually have 1/3 of that toxin and still have more efficacy if you engineer these ADCs precisely. Okay, so at this point, we are having a library of these ADCs. We already have screened them to the cellular level, then shortlisted a few of them that are currently being tried in vivo, along with some other preclinical data to see what we can take forward. And then, as JD mentioned, we are in the process of signing a contract with one of the biopharma companies, which is currently testing one of the ADCs to see how they will be establishing the workflow, because they have the expertise from preclinical to the clinics, and that's where we are working with them. Now, there's one last aspect related to this that I'll discuss today, and then I'll close this part of the story. So far, we were discussing about using lysines or those blue residues as linchpins, and they're great till you are working with isolated proteins. Till you are dealing with 50 people, it's easy to find that combination where a male sitting at a defined distance from a female would still give you a unique pair. But if you're dealing with 10,000 people, then the same restriction might give you more than one combinations that would work, right? So it would probably be giving you, you will have to put another filter to it, or you will have to change the characteristic of either the male or the female, right? So think about it. If I kind of redefine it as a male sitting at a defined distance from a female with a red hair, then in that case, this becomes more restrictive because the number of females with red hair would be very limited, right? So in terms of protein modification, it's as simple as saying that instead of taking something that is of high frequency for guiding the modification, how about we take something that is of low frequency? And then if I'm working with 20,000 proteins simultaneously, I might still be able to find a unique pair for a unique protein and target it specifically. So that is exactly what we did. We know that cysteine is a low frequency residue. We use that as the linchpin for directing the modification of a particular protein. So we were able to demonstrate that you can do a single site modification of a single protein within a complex cell lysate. It also, along with that, allows you to access a completely new targetome for making the antibody drug conjugates, but I have already shared that part of the story. Just one last question, and I'll wrap it up. Two equilibria or two reversible reactions is what we talked about. One question that we were investigating for almost 7 years now was, can we have a complex multi-equilibria system like what the biological systems have? Would it be possible to have one specific pathway giving one product at a time like what biological systems do? So 5 minutes. So it's as simple as that can I have very specific substrate, and that substrate would lead to a particular response. One signal, one response, another signal, another response. And the answer is yes, that's possible. And we can take a multi-equilibria system, you leave the details. The important point is you can have one very specific pathway leading to one specific carboxylate modification. You can change the signal in terms of reagent and the pathway changes, and it happens completely exclusively. You can switch from pathway 1 to 2 to 3 to 4, and that works pretty well in delivering some of the chemistries that we did not imagine in the past. And the interesting thing right now is that this is one of the platforms that we have scaled up to multi-milligram scale. And from the biopharma perspective, that's pretty good scale for all the preclinical investigations. Now, I hope that I was able to convince you today that human behavior is a great model system for looking into some of the complex problems that are having multiple variables, such as precision engineering of proteins. And it led to the disintegrate theory for us, which allowed us to simultaneously regulate reactivity with chemoselectivity, site selectivity, protein specificity, modularity, and so on. And one part of our group has been working for last 6 years or so with trying to see if we can target proteins specifically within the live cells and the living systems And that's a story that probably I'll be sharing at another time. But another part of the group is looking into how the selected ADCs would perform in vivo. And do I have 2 minutes or should I? Okay. So I'll just show you one last video on what Playbill Tech is doing in one of the workflows, apart from translating everything else that I showed today. And let me see if it has an words.
Cancer is the second leading cause of death worldwide. Patient survival benefits from complete and precise surgical tumor removal. However, surgeons often rely on unprecise techniques such as inspection and palpation to distinguish between tumor and healthy tissue. Due to this challenge, 6% of colorectal cancer resections are incomplete. Our group aimed to help surgeons detect tumor tissue by developing fluorescent tracers. These tracers bind to cancer-associated proteins on tumor cells. Members of the F receptor family are specifically upregulated in colorectal cancer, whereas it is almost completely absent in surrounding normal tissue. Based on our research, we are developing tracers targeting the F receptor for fluorescent image-guided surgery in order to assist surgeons in identifying colorectal cancer. Image-guided surgery uses tracers consisting of antibodies or peptides coupled with fluorescent dyes which bind to proteins like the F receptors. When the tracer is excited by near-infrared light, it emits fluorescent light, which can be detected using dedicated camera systems. After excitation, a fluorescent signal is seen on the surgical video monitor, indicating the presence of tumor tissue. Using this technique, the boundaries between the tumor and healthy tissue are clear. The surgeon can then remove all of the tumor, resulting in improved patient outcomes.
Okay, so with that, I guess it's time to thank the actual scientists who did all the work that I discussed today. The amazingly talented group of people. You know, I would really have a high regards for all this first generation of students, because they started working on these projects when we were not sure if anything would work out or not. But then the current generation of students are also taking a lot of challenging questions forward. But I must thank all my collaborators. You know, all this multidisciplinary research that I share today, it does not come just from my lab. We have a close-knit group of people who work together, and depending on the scientific challenge that we are addressing, whether it's Ram, Sanjeev, Devashish, or Saptarshi, or Suman, or Chris, or Avinash, or Vatsan, or Akansha, they contribute in different dimensions. I was able to share only some part of these stories where some of these people have contributed. The other part I was not able to share it today. We do work actively with multiple biopharma companies across the country as well as outside the country. And I can't thank the funding agencies enough, because unlike TIFR, we do not get a lot of intramural research grant. So we have to depend a lot on the funding agencies for their generous support for our research over all these years. Thank you very much. And I'll be glad to take any questions that you might have.
Thank you. This was fantastic. Thank you for this wonderful introduction to the ideas and also the research. So, questions for Vishal? Yes.
Yeah, so I have a question about the ADCs. So when you sold the ADC, that was one was conjugated to doxo, another one I just forgot. So what was the release mechanism from that linker?
That's a very good point. So let me just, so one was DM1 or, okay, let me see. Right. So in this particular case, we normally don't talk about the linker chemistry at all. The ones that we are right now taking in vivo are the ones where we have very specific linkers. Linker technology is not from our lab. That is something that we have taken based on what is out of patent and what will give us the fastest route to market, because our novelty is not coming from the linker or the toxin or the antibody. It's based on the control over the precision. So we can talk about it off record because some of those things I won't disclose.
Yeah.
But in this chemistry, this is good enough. So this carbon-nitrogen bond breaks pretty conveniently. It is great for cellular studies. I would never recommend it for in vivo studies, but this is the one that I would still recommend for in vivo studies. These are non-cleavable linkers. It will be protease-mediated degradation. You do not have a linker that is going to be cleaved specifically from a protease in this case. However, the ones that we are trying in vivo right now are the ones that do have a protease-specific linker. So there are two filters. One, the antigens on the tumor. The second, the protease overexpression for the cleavage of that specific linker. So I hope you got the point.
Yeah.
So that increases the specificity and reduces the off-target toxicity.
And just one more thing. So for these kind of conjugates antibody, where you have this artificially designed link, how does the immunoreactivity affects the
How does the
immunoreactivity of the antibody? So if you compare
are
with
they-
the native antibody, so does it lose certain percent? And
That's
what is
again
the
an
thing?
excellent question. So that is exactly the problem why the antibody conjugates that are made heterogeneously, their activity drops down substantially, and the dose has to be increased quite a lot to compensate for it. Because the part that I did not discuss at all, I said that in the Y, the top parts are involved in engaging the antigen, but the fit part is something that engages the immune cells, right? The problem with many of those heterogeneous conjugation is that they target even that. So your regular capabilities of engaging immune cell to kill the tumor cell is gone because of that. So in these cases, we shortlisted and I showed only the ones where we are not touching either the top of the Y or the bottom of the Y. But we do have those examples as well, and we do see effect on the ADCC, the antibody-dependent cellular cytotoxicity.
Okay. I have a question, Vishal.
Yes.
How many now you can get with these kinds of toolkits that how many toxin loaded per antibody and a drug loaded per antibody?
So
Can you actually get with one?
we have
one
made it
tagged?
work one.
Okay.
Yes.
Okay.
But that is not the most efficient one.
Acha. So you
we-
might want to
Yeah.
increase the load.
Right. So we realized that putting it somewhere between 2 to 3
Okay.
is going
Is the most lethal one.
Yes, so it gives you enough dosage of toxin.
Yeah.
But we still do not know in the long run with in vivo studies.
Whether they are stable or
No, they are stable.
stable.
But would they still be the best performers, or we will have to come down to the one where we are putting only one?
Okay.
Can I
Okay.
ask one question? Yeah. So now there are basically two parameters, I think I can see. So one is the circulation time of the antibody.
Yes.
That is very important.
Yeah.
Yes.
And
I'm glad that you
second-
pointed it out.
Second thing is that the ultimate local dose of the
Mm.
toxin that you need to kill the cells.
Right.
Mm.
Right. So if I have to think about putting the two
Both.
things together, how do I decide the ratio?
Right. Perfect question to ask. Now, this is where we were lucky to actually also get the collaborators to work on antibody fluorophore conjugates. Because while we were talking to these surgeons, we realized that right now they use the exact technology that is used for making antibody drug conjugates, and they get it through one of the companies constructed for them. And the problem, so the advantage is that with antibody fluorophore conjugates, you can also image what is happening with the clearance rates and non-specific localizations. So that actually helps us having a better understanding for what is needed in the ADCs as well. Now, in this case, it is quite obvious that when you're putting these decorations heterogeneously, they will offer you a spectrum of behavior. Their distribution and excretion is not going to remain similar for different chemical entities. The polarities and the surface charges and everything is changing with each entity, right? So that is a problem. And you would realize that if you're loading it too much, it's faster clearance. That's again something that's not required.
But how
But-
this is possible? Your loading is too much.
Yeah, because all these toxins that you're putting in, they're quite hydrophobic in nature. Now, the argument that they put in is that this whole hydrophobic chain that you're putting in is somehow responsible for faster clearance. But it is noticed that these things are cleared faster. Having said that, I have also seen data where the reverse is true, where the argument is that these non-specific localizations take longer to clear off. So for the surgery purpose, unless these non-specific localizations are cleared off, you will have to wait. And while you are waiting, after the injection of these AFCs, the ones that are bound to tumor start getting internalized. And when that happens, you are losing the observation for margins, which is the most tricky part to actually see, and you might end up leaving certain cells.
That
you
is
don't-
what I wanted to ask you afterwards.
Yeah.
So, but the thing is then, how do I kind of rationally think about, let's say if I know the killing ability of a particular killer, and I know the IC50, which is, let's say, in cells or even in animal, I know the dose. Okay,
Yes.
this much dose
Yes.
gives me this much of local dose.
Yes.
Then I want to put the drug so that nothing goes anywhere and every dose goes to there.
So antibody behaves like antibody and does not give a diverse, spectrum of
Yeah.
behavior. That is what, if you want,
Yeah.
then you would want to probably homogeneously attach these toxins or whatever payload you want to put in at the hinge regions close to that, and not touch the fab part, not the bottom part. And it would not be a problem till your loading is between 1 to 4 drugs. Okay. That is assuming you have not disturbed the structure of the antibody by reducing the disulfide and use that approach.
But you're using some other tagging technique, right?
So if
you're not
you're-
using
yeah.
cysteine.
So if you, well, in one chemistry, we
Okay, you-
use that also to just see what's the whole fuss about and why people who are working in that field are working on it. I would not say anything officially about how bad this could be.
Okay.
Yeah, but if you reduce the disulfide,
Yeah.
you will definitely not
a
have
problem.
the same rebridging.
Sure. Okay.
Thank you.
Okay. If there are no more questions, thank you for sticking around for the question answer session. We will move to the snacks area and let's actually have some coffee and tea with Vishal. So thank you. Thank you very
Thank you
much.
very much, guys.
I thank the auditorium staff for keeping up with the Zoom and helping us with the broadcast. Thank you.